Today in biology class 6, I'm going to present the basics in basic difference in between the animals and plant cells. So as we are familiar that there are fundamental similarities, but still there are differences between plant and animal cells. So if we are talking, for example, the animal cells which have centrioles, centrosomes, and lysosomes, which is completely absent in plant cells. And plant cells have cell wall, chloroplasts, plasmodesmata, and plastids, which are in general used for storage and a large central vacuole where if you want to see and know in animal cells, these are not present at all. So this is some of the listed differences, and I have already explained the comparative study or comparison between prokaryotic and eukaryotic cells. So for example, if we are talking some examples of prokaryotes and eukaryotes, here you can see bacteria in prokaryotes and eukaryotes, fungi, plants, and animals. The organization level in prokaryote is single cell, while eukaryotes are from single to multiple or multicellular. Typical sizes are also different in plant eukaryotes and prokaryotes. Cell wall, almost all the prokaryotes have cell walls, while eukaryotes here, fungi and plants have cell walls, but animals do not. Organelles normally prokaryotes do not have, while eukaryotes have. And there are some metabolic differences like anaerobic and aerobic diverse. But in eukaryotes, mostly aerobic metabolic activities could be observed. In terms of genetic material, single circular double-stranded DNA is present, while in eukaryotes, you could see well-defined nucleic acids DNA. Mode of division, binary fissions, mostly violet, such as like here you can also see budding, but in case of eukaryotes, you can see the proper cell division processes like mitosis, meiosis. Now, I'm coming one by one for some of the cell part or components which I have not introduced. And first one is cell wall. As we have learned now that plant cells have the cell wall. And the cell wall could be seen here in this plant cell that is here, just above plasma membrane or cell membrane. And this is like if you want to see in terms of well-defined structures. So this is the right side is also similar thing just to represent in well-defined way. So for the cell wall, for example, you could see here primary cell wall, secondary cell wall, and then something called middle gap, that is middle lamella. Then these composition cell wall structure could be like primary cell wall, which you can see here from inside, and then secondary cell wall is here, like this is from this part. So if you could see the lining, how it is indicating the primary and secondary cell wall. And then the white gap is coming here, that is middle lamella. So let's see here, the line indicates that secondary cell wall, here it is inside the lining, And then primary cell wall is this green structure. And in terms of composition, you can see here like pectin, cellulose, microfibril, hemicellulose. And then if you want to know what comes just after cell wall, then it is plasma membrane or cell membrane. Next, it is chloroplast. This is also very important component of plant cells. Similar to mitochondria, chloroplasts have their own DNA and ribosomes, and it function in photosynthesis and can be found in eukaryotic cells such as plants and algae. Then, like in photosynthesis, what happens, what is the role, which you can see when we will be discussing about photosynthesis in detail. So here CO2, like carbon dioxide, water, and light energy are used to make glucose and oxygen. And this is the major difference between plants and animals. So plants which are like able to make their own food, like glucose, whereas animals must rely on other organisms on their organic compounds or food source. There are two things written in bracket. In case of plants, it is written autotrophs, and in animals, it is written heterotrophs. So these are some of the keywords which you must need to know what in exact autotroph means, and these are some of the quick questions which are asked in exams. So similar to mitochondria, chloroplasts have outer and inner membranes, but within the space, as I mentioned, it is inner membrane, which is like, a set of interconnected, stacked fluid cell-filled membrane sacs that is called thylakoids. Each stack and thylakoids is called a grana, that is the fluid enclosed by the inner membrane and surrounding the grana is called stroma. So these are some of the things which you need to remember, like in intermediate membrane, outer membrane, inner membrane, granum, stroma, thylakoid. So this is the diagram which represents the simple diagram for a chloroplast with the membrane details and some of the major components. The chloroplasts contain a green pigment called chlorophyll. And this is like several times repeated questions are coming on chlorophyll, which captures the energy of sunlight for photosynthesis purpose. Like plant cells, photosynthetic protists also have chloroplasts. Some bacteria also perform photosynthesis, but they do not have chloroplasts. So their photosynthetic pigments are located in the thylakoid membrane within the cell itself. Now, there is a term which came just during this study, that is endosymbiosis. So what is endosymbiosis? It is a kind of theory that attempts to explain the origin of organelles, mitochondria, and chloroplasts. There are three kinds of endosymbiosis: primary, secondary, and tertiary. So symbiosis is quite clear. The people could understand about symbiosis. But there is some interesting thing which you need to know, why we are interested to know the endosymbiosis and why it came here. So you could understand from the right side figure, where you can see even not just right side figure, but also in the middle figures, some plant components are here. And that is like endosymbiosis primary is a key evolutionary process where eukaryotic cell engulfs a prokaryotic cell, leading to the formation of mitochondria and plastids. So this event is considered as the origin of these crucial organelles in eukaryotic cells. Then secondary endosymbiosis is a process where a eukaryotic cell engulfs another eukaryotic cell that already contains a chloroplast. So here you can see secondary endosymbiosis. This was primary endosymbiosis, where you can see clearly how the engulfing process is taking place. And then there are something called gene loss. And then there is a new kind of originated species. So then it comes tertiary endosymbiosis, which you are seeing here. So tertiary endosymbiosis, which is basically a type of endosymbiosis where a eukaryotic cell engulfs another eukaryotic cell that already possesses secondary plastid. The next part is the central vacuole. So as you can see in any plant cell, there is a big vacuole, which is called as central vacuole. Other than the cell wall, cytoplasm, cell membrane, cell sap, tonoplast, you can see a big structure inside the cell that is a vacuole. So the vacuole here is like a kind of gap, you can see. And if you want to understand, like normally plant cells have a large central vacuole. And what it contains? A lipid called cell sap. And cell sap contains dissolved substances such as sugars, minerals, salts, and amino acids. What is quite important here? What is the central role or key role? So its key role is in regulating cells' concentration of water in changing environmental conditions. So now you can understand why it is important and what is the significance of presence of central vacuole. Now it comes to ECM or extracellular matrix, which is present in animal cells. So of course, this from here it looks like something small granules, and simple things. But these days, extracellular matrix or ECM is very important, especially to understand cancer cell biology. So those people who are expert of pure cancer biology, they are highly interested in understanding extracellular matrix or ECM. So what it is, most animal cells releases materials into this extracellular space. The primary components of these materials are glycoproteins and the protein that is collagen protein. So these materials are together called extracellular matrix. So that means collagen protein and glycoproteins together are ECM or extracellular matrix. Not only does the extracellular matrix hold these cells together to form a tissue, but it is also allowing the cells within the tissues to communicate with each other. So you could understand from this figure, the right side figure, how the roles of ECM could be seen in cell or tissues. So first thing it comes is kind of to maintain the stiffness of the cell or to maintain the real shape and position of cell and cell organelles in the proper or appropriate place. Second thing is cellular communication. So here you can see endothelial tissue, then basal lamina, then connective tissue, then collagen fibers. So what it says here, mechanical stresses are transmitted from cell to cell. That means cellular communication by cytoskeletal filaments anchored to cell matrix and cell-cell adhesion sites. Now, extracellular matrix directly bears mechanical stresses of tension and compression. So there are enormous roles and significance of ECM or extracellular matrix in animal cells, not only in cancer, but also in several other diseases. So that is all for today, what I wanted to say. And in conclusion, in the end, what I'm saying here, showing here, is like the real extracellular matrix composition in terms of a network, substances secreted by cells, carbohydrate, protein, polysaccharides, fibronectin, collagen fiber, integrin, microfilaments of the cell. So these are the things which you might be seeing when you are trying to understand deep biology. So blood clotting provides an example of the role of ECM in cell communication. When the cell lining a blood vessel are damaged, these display a protein receptor called tissue factor. When tissue factor binds with another factor in ECM or extracellular matrix, it causes platelets to adhere to the cell wall, to the cell, sorry, of the damaged blood vessel, thus stimulating adjacent smooth muscle cells in the blood vessel to contact. That means contract. So, or initiate a series of steps that stimulate the platelets to produce clotting factors leading to blood clot. And that's all. So next class will be on intracellular junctions. So thank you very much.